Assalamualaikum. The topic of this lecture is molecular basis of cancer. We will be doing this in parts. Molecular basis of cancer will be covering some important terminologies related to carcinogenesis. And in molecular basis of cancer part 2, we'll be doing uh, some tumor suppressors. These are our learning objectives for molecular basis of cancer. One, now, there is a genetic hypothesis of cancer which suggests that the tumor results from the clonal expansion of a single progenitor cell that has undergone some genetic damage. This damage may be inherited, acquired, or spontaneous. So initially, all tumors, they are copies or clones of a single mutated progenitor cell. That is why they are monoclonal in origin. Now, there are four classes of normal regulatory genes, which are the principal targets of cancer. These include growth promoting oncogenes, growth inhibiting tumor suppressor genes, genes involved in the repair of DNA, and genes that regulate apoptosis. Before we move on further, we'll discuss the important types of mutations, gain of function mutations, and loss of function mutations. Gain of function mutations are those mutations in which the gene either gains a function or its function is amplified. So these type of mutations, they can amplify the expression of genes or result in a new function of the gene. Whereas the loss of function mutations, they lead to deletion of the gene sequence and therefore the function of that gene is lost. Now, proto-oncogenes, these are genes that normally help the cells to grow and divide, to make new cells, and to stay alive. So these are necessary for growth and survival for the cells in the body. Oncogenes are formed when there are mutations in proto-oncogenes. The proto-oncogene is a normally regulatory gene, and when it is mutated, it becomes an oncogene. Proto-oncogenes may be growth factors or growth factor receptors. They may be transcription factors. They may be signal transduction proteins, or they may be proteins that inhibit apoptosis. There are different types of proto-oncogenes. Oncogenes, they can be transcription factors, they can be growth regulating proteins, or proteins involved in cell survival, or proteins involved in cell-to-cell interactions. Oncogenes are dominant because mutation of a single allele leads to transformation of that cell into a cancerous cell. A proto-oncogene, when it acquires gain function mutation, it becomes an oncogene. An oncogene, even if a single allele is mutated, it will have a dominant effect and lead to cancer. Oncoproteins are those proteins which are included by oncogenes. They promote cell growth in the absence of growth promoting signals. We discussed earlier that there are four classes of regulatory genes. We'll be discussing what happens when mutations occur in these genes. So first, mutations activating growth promoting oncogenes. These type of genes, the growth promoting oncogenes, they undergo gain of function mutations, and even if a single allele of the gene is mutated, it has a dominant action and it transforms the cell into a cancer cell, into a tumor cell. Next is mutations that affect the tumor suppressor genes. The tumor suppressor genes in the body, they are responsible for suppressing the growth of tumors. How do they suppress the growth of tumors? By uh, initiating apoptosis of those cells which have undergone DNA damage and they are not able to repair that damage, or cells which have undergone genetic mutation, they are stopped from moving into the cell cycle. When tumor suppressor genes, they undergo mutation. These type of mutations are loss of function mutation. So the function of tumor suppressor genes, which is inhibit the growth of cells that is lost, and these type of mutations, these type of genes, they require two mutations in both the alleles for the function to be, for the function of the tumor suppressor gene to be lost. So here you can see in the picture, there is a normal cell. When it undergoes one mutation, it activates the tumor suppressor allele, uh, inactivates the tumor suppressor gene, and only a single allele is affected. But when the second mutation occurs and both the alleles, both the genes, both the alleles on the gene are affected, then the tumor suppressor gene, it loses its function. This is a recessive effect. In order to understand the concept better, you can see there is a picture of a car. So the car has an accelerator and it also has a brake. You can think of an accelerator as a gene which is mostly off and only switched on when growth of cell is required. Tumor suppressor genes, they are in working condition. They act as the brake of the cell cycle. So when the genes of the tumor suppressor genes and oncogenes, they are mutated, the brake becomes defective and the accelerator, it is jammed. So the accelerator, it remains pushed, which are oncogenes, and they help the cell to keep on going, whereas the tumor suppressor genes, they have lost their function, so the brake has undergone damage, and it cannot stop these mutated cells or damaged cells from growing. There is uncontrolled growth of the mutated cells. Third is mutations occurring in apoptosis regulating genes. When apoptotic genes, the anti-apoptotic genes, they undergo mutations. What is the result? There is less death and enhanced survival of the cells. Apoptosis or damaged cells does not occur, which leads to their survival. Next, we have mutations that affect the DNA repair genes. These type of mutations, they impair the ability of the cell to recognize and repair damage in other genes. And that is why cells which have the damaged DNA or damaged genetic material, they keep on proliferating. They are not repaired and they do not undergo apoptosis. Carcinogenesis, it is the formation of cancer. Formation of cancer or the formation of the tumor, it is not a single step. It is stepwise accumulation of mutations over a period of time that results in cancer. The normal cells are eventually transformed into cancer cells once they have undergone a series of mutations, once they have accumulated a number of mutations stepwise. Carcinogenesis results from accumulation of mutations in stepwise fashion. And these mutations, they are complementary, they help each other climb the ladder towards formation of cancer. Now, there are two other important types of mutations, driver mutations and passenger mutations. Driver mutations are those mutations which drive the cell towards carcinogenesis of cancer formation. They have phenotypic effect on the cancer cells. Passenger mutations are just like an audience or the passengers of the bus. They do not alter the phenotype of the cancer cells. Here you can see in the picture, the capital letters, they are indicating the driver mutations which have an effect on the phenotype, whereas the passenger mutations, they just carry along, they do not have any effect on the phenotype of the cancer cells. As we discussed earlier, initially when a tumor is formed, it is composed of copies of roots of a single mutated progenitor cell. Initially, a tumor is monoclonal. However, as it gradually accumulates more than one mutations, it evolves and later on it becomes heterogeneous. In this picture, you can see all the tumor cells initially were blue in color. Later on, when they evolved by undergoing more mutations, the color has started to change, and the final tumor is composed of three types of cells: yellow, green, and red. So it has become heterogeneous. Now, cancer hallmarks are the phenotypic attributes of malignant tumors, which occur due to genetic mutations or changes in the genetic material. For example, excessive growth, local invasiveness, and the ability to form distinct metastases. These are the phenotypic properties of cancer or the cancer hormones. All the cancers, they display some fundamental changes, and these are called as hallmarks of cancer. Here in this picture, you can see the different hallmarks of cancer: invasion and metastasis, inflammation, increased replicative immortality. They keep on replicating and will not stop. Angiogenesis, formation of the blood vessels so that they can acquire more immune nutrients. Sustained proliferative signaling. They keep on giving signals for proliferation. They do not require any kind of growth factor for their proliferation and growth. Then cancer cells, they also evade or avoid the immune destruction. They have metabolic reprogramming, which is Warburg effect. They are resistant to cell death and apoptosis. They also bypass or evade the growth suppressors, and they are genomically unstable. These are the properties of cancer cells. Now, how do normal cells respond to growth factors? Normal cells usually require a growth factor to attach to its receptor in the cell membrane that the signaling molecules can be stimulated within the cell, and then the signaling molecules activate the transcription factors within the nucleus, which starts the replication of DNA. However, in cases of cancer, they do not require any growth factors. They are self-sufficient in their growth signaling, and they keep on proliferating in the absence of any growth factors. Many of the oncogenes, they include growth factor receptors like receptor tyrosine kinases, which have independent activity. They are not dependent on the growth factors for their growth. These are the major signaling pathways which are affected in various cancers. These include receptor tyrosine kinase pathway, which is the most important and most common. Then G protein coupled receptor pathway, this is also affected. JAK-STAT pathway, WNT pathway, NOTCH pathway, HBOC pathway, nuclear factor kappa B pathway, transforming growth factor beta SPAD pathway. These are various pathways which may be affected during carcinogenesis, and the most common of these is the receptor tyrosine kinase pathway, which is a growth stimulating pathway. Here you can see normally this requires a growth factor to be activated, but in cases of cancer, no growth factor is required to stimulate the receptor. The receptor itself is abnormal and it is self-sufficient in growth signaling. Now, cyclins and cyclin-dependent kinases, they are also important in carcinogenesis because they drive the cell cycle. The growth factors, they stimulate the progression of cells through the cell cycle by using these cyclins and cyclin-dependent kinases. You can see in the picture, the various phases of cell cycle, they are dependent on cyclin-dependent kinases and the cyclins. Now we come to the classification of oncogenes according to their mode of action and the tumors that are associated with these. Oncogenes can be growth factors like platelet-derived growth factor beta, which if overexpressed can lead to astrocytoma. Fibroblast growth factor is another oncogene which is overexpressed or amplified in various tumors like osteosarcoma, stomach cancer, bladder cancer, melanoma, and breast cancer. Transforming growth factor alpha is overexpressed in astrocytosis, which is a brain tumor. In hepatocyte, growth factor is overexpressed in hepatocellular carcinomas and thyroid cancer. Oncogenes can be growth factor receptors, and the important growth factor receptors are epidermal growth factor receptor family, which is mutated or amplified in adenocarcinoma, lung and breast carcinoma. Platelet-derived growth factor receptor is overexpressed or translocated in gliomas, which are brain tumors, and leukemias, which is blood cancer. ALK receptor may be mutated in cases of adenocarcinoma, lung, some lymphomas, and neuroblastoma. Oncogenes may also be signal transduction proteins, which include GTP binding proteins, non-receptor tyrosine kinase, BRAF, and JAK-STAT or JAK2. And when they undergo mutation, they are associated with various tumors. Similarly, oncogenes may also include for nuclear and cell cycle regulatory proteins like cyclins and transcription proteins like MEK and N. When they are mutated, they can lead to various tumors like lymphomas, brain tumors, melanomas, esophageal cancers, breast cancer, and sarcomas. Now coming to the part 2 of molecular basis of cancer. These are the learning objectives. We'll be discussing tumor suppressor genes mainly. Now, tumor suppressor genes are those genes which suppress the formation of tumor. How do they do that? They recognize genotoxic stress. They apply brake to the cell cycle. They control various checkpoints within the cell cycle, and they prevent malignant transformation. Immunosuppressor genes include proteins that function as transcription factors, cell cycle inhibitors, signal transduction molecules, cell surface receptors, and regulators of DNA damage. Just like we classified oncogenes previously, we also classified tumor suppressor genes based on the mechanism. Tumor suppressor genes may be inhibitors of growth signaling pathways like APC gene, NF gene, PTEN, and SMAD. They may be inhibitors of cell cycle progression like retinoblastoma and cyclin-dependent kinase 2A gene. They may be inhibitors of growth metabolism and angiogenesis like VHL, STK11, and succinate dehydrogenase. They may be inhibitors of invasion and metastasis like cadherin gene. They may be enablers of genomic stability like TP53. They may be DNA repair factors like BRCA1, BRCA2, and mismatch genes, or they may have an unknown mechanism of action, which include WT1 and menin gene. These are all tumor suppressor genes. They have been classified based on their mechanism. Now, Knudsen was an American physician and genetist who gave a two-hit hypothesis. He stated that most of the tumor suppressor genes they require mutations encoded because of phenotypic genes. If you remember, in the start of the lecture, we discussed that loss of function mutations occur in tumor suppressor genes, and in order to lose its function, the tumor suppressor gene must undergo two mutations because it is recessive. So this is the two-hit hypothesis. Retinoblastoma gene is a tumor suppressor gene. 98% of retinoblastoma cases, the RP1 gene is inactivated. Retinoblastoma may be hereditary or it may be sporadic. In cases of hereditary retinoblastoma, the first somatic mutation occurs before birth and the next mutation occurs after birth. So when two mutations have occurred, then retinoblastoma occurs. While in case of sporadic retinoblastoma, both the mutations they are acquired or gained after birth, and these are loss of function mutations. The first loss of function mutation, when it occurs, the function of the immunosuppressor gene retinoblastoma, it is decreased, and when the second mutation occurs, the function of retinoblastoma gene is completely lost. So carriers of retinoblastoma mutated gene, they have a 10,000-fold increased risk of developing bilateral retinoblastomas compared with the general population. Retinoblastoma gene is the governor of proliferation. It is also called as the governor of proliferation, and it is a negative regulator of the G1 and synthesis cell cycle transition. So there is a checkpoint in the cell cycle, which is called as G1S, and this is the place where retinoblastoma gene acts. Normally, it exists in an active hypophosphorylated state in the resting cells, and in this state, it is bound to E2F, which is a transcription factor. When this gene, retinoblastoma gene, it is hyperphosphorylated, it becomes inactive, it releases E2F, which is a transcription factor, and because the transcription factor is free, it progresses the cell cycle. Here again in this picture, you can see that retinoblastoma, when it is active in resting cells, it is bound to E2F, which is a transcription factor. When retinoblastoma gene is phosphorylated by cyclins, it becomes inactive. Retinoblastoma gene, when it becomes inactive, it releases E2F. E2F then acts on the nucleus of the cell and starts the cell cycle. So the cell starts to grow. In cases of cancer, retinoblastoma gene, it loses its function. It becomes hyperphosphorylated and it releases the E2F transcription factor for the cell to grow. The anti-proliferative effect of retinoblastoma gene is overcome by a variety of mechanisms. Loss of function mutations can occur in this gene. Cyclin genes or cyclin-dependent kinases may be amplified. When cyclins and or cyclin-dependent kinases are amplified, they hyperphosphorylate retinoblastoma gene or make it inactive. So when the tumor suppressor gene retinoblastoma, it is inactive, it cannot perform its function of suppressing the tumor. Other mutations can also occur in cases of retinoblastoma gene. These include loss of function mutations affecting P16. P16 is an inhibitor of cyclins, which are inhibitors of retinoblastoma. Viral oncoproteins like E7, it also binds and inhibits retinoblastoma gene. So when retinoblastoma gene is inhibited, it allows the cell cycle to progress. Another important tumor suppressor gene that we'll be discussing in this topic is DP53, which is called as the guardian gene. It regulates cell cycle progression, it repairs DNA, and this gene also leads to cellular aging and apoptosis of damaged cells. This is the most frequently mutated gene in human cancers. Loss of function mutations occur in this gene. Lee-Humani syndrome is a syndrome associated with mutations in this gene. Individuals usually inherit one mutated TP53 allele, and that is why they have 25-fold greater chance of cancer. So one allele, it already has the mutation at birth, and then after birth of the individual, the other mutation, it is acquired and it leads to Lee-Humani syndrome. Now, this TP53 gene, it has an important role in preventing cancer development. In normal cells, it is undetectable. TP53 is not detectable in normal cells. When there are stressful signals like DNA damage, shortened telomeres, hypoxia, or genetic mutations, P53 is released from the inhibitory effect of MDM2. When P53 is released, it can either arrest the cell cycle, it can initiate DNA repair, or it can, if the repair cannot occur, it will allow the cell to undergo apoptosis. So the E6 protein of high-risk human papillomavirus, it binds P53 and it promotes its degradation. So when this tumor suppressor gene is lost in case of human papillomavirus, it can cause cervical cancer and head and neck cancer. So the role of P53 in inhibiting neoplastic transformation is by inhibiting the cell cycle transiently or permanently, or by allowing the cells to undergo apoptosis. When P53 is lost, cells cannot repair the damaged DNA, driver mutations start to accumulate, and finally carcinogenesis occurs. Now, this is a table that is showing various types of tumor suppressor genes along with the mechanism of these genes and the associated tumors. So the APC or adenomatosis polyposis coli gene, this is associated with colonic cancer. In fact, the E-cadherin gene is associated with familial gastric carcinoma. Cyclin-dependent kinase 2A gene, this is associated with melanomas, familial melanomas, and leukemias and transformers. Transforming growth factor beta is another tumor suppressor gene that is associated with the tumors of GIT and endometrium. P10 is associated with the risk of breast and endometrial carcinoma. VHL1 gene is associated with renal cell carcinoma and pheochromocytoma. These are all tumor suppressor genes which, when undergo loss of function mutations, their function is lost, and therefore the damaged cells or the mutated cells they keep on going without any check and balance. This is all. Thank you.